Thanks so much, Jeremy, and thanks to JGI and particularly the plant group for all the enabling work that you guys have been doing. So we're going to take a brisk climb through the green tree of life, and I'm going to be just giving you a little kind of scratching the surface on a number of stories that we've been working on quite a bit now, more and more with the plant group at JGI. So first of all, folks probably you've exposed this first quote from Dobzhansky in '73. Nothing in biology makes sense except for in the context of evolution. Many folks that are making phylogenies, both gene phylogenies and species phylogenies, would say nothing in evolution makes sense except for in light of phylogeny. And this was evident in the way that Charles Darwin was thinking about the diversity that he was experiencing as he's sailing around in the Beagle and seeing this diversity and sketching in his notepad and saying, well, these things all look alike. To what extent is this due to shared ancestry? Jump ahead now, and we're in the era of phylogenomics, and we're using genome scale data to reconstruct relationships among species, and then using that understanding of species relationships to understand the evolution of genes and gene families and genomes in that context. So I'm going to talk just briefly about the first part of this, that using big genome scale data, this transcriptome data set that Jeremy mentioned, to reconstruct relationships among species. And this is work that we were looking at reconstructing relationships across Viridae plantae. There's the workflow: grind up some material, make sure the RNA looks okay, run it on a Luminous sequencer, assemble the transcriptomes for 1,000+ species, and then we have to organize all of those transcripts that we have assembled into gene families, and tools are now available for doing that quite well. And then we've got thousands of gene families and 1,000 or so species that we wanted to make multiple sequence alignments for in gene trees. And this is a massive undertaking, and we needed help from computer scientists to be able to do this sort of work in a very efficient way. And so Tandy Warnow, Siavash Marab, and Anam Pham were instrumental in helping us come up with ways to rapidly make these multiple sequence alignments in gene trees, and then use those gene trees to reconstruct species trees. Well, I'll figure this out at some point. So here's the tree from the 1KP project. And there's a couple things that I think are really important here. One is we've kind of resolved some relationships that had been difficult for a long time for molecular systematists to resolve. You're largely using chloroplast genes and genomes. But actually something that's more interesting to me in some ways is the variation in the branch links. And these are this variation in branch link is due to time since speciation events and effective population size of this ancestral species. So if you've got a large ancestral species with a large effective population size, lots of allelic variation, that allelic variation is being pulled through multiple speciation events, and leading to gene tree, species tree discordance. We use coalescence approaches to dealing with that and reconstructing relationships. So, so this was a very interesting finding and a big undertaking involving a large community of folks. This work is being carried on using the same loci that we really focused on, single copy, about 400 single copy genes that seem to be shared across the Viridae plantae. This work is being carried on I said 353 of those genes. Folks at Q and collaborators have made gene capture kits, and they're using this gene capture approach to try to reconstruct relationships among all genera in flowering plants in particular. And so in phase one, they've gotten to 60% of genera for flowering plants, and now we're working to move on to get the rest over the next 4 years. But in the meantime, if you want to go check out that website, and it has a tree that you can explore and understand the relationships of genera, you know, good number, 60% of the genera at this point anyway, across the flowering plant phylogeny. Okay, so now we've got an organismal relationship, and we want to use that understanding to investigate gene family and genome evolution in the context of that organismal phylogeny. And you know, phytosome and a lot of these databases are wonderful and so useful. That with this avalanche of data that's coming at us now, with new genomes coming out every day, really, we really are working on trying to develop pipelines that folks would be able to use and update or focus in on parts of the tree of life or particular gene families with data that's coming out, again, on a daily basis, new data coming out on a daily basis. This is involving, again, clustering of genes in the gene families, doing multiple sequence alignments, making gene trees, and then refining those gene trees with SNP data and our understanding of species trees. Okay, I'll get this yet. All right, so now I want to talk a little bit more about how we can use this sort of data to understand genotype-phenotype relationships. And the way that we can do this most efficiently is, as was mentioned in Igor's talk, is by comparing very closely related species or genotypes within species. And so I'm going to give a couple vignettes where we've done this. So first of all, I'll talk a little bit about the evolution of dioecious and sex chromosomes in plants, the flowering plants particularly. About 6% of flowering plants are dioecious and have separate sex chromosomes, including Amborella, which is sister to the rest of the flowering plants. Now, Amborella, it is not, it's a sister lineage to all other flowering plants, but dioecious is a derived trait in Amborella, and this dioecious has arisen multiple times, hundreds of times, across flowering plant evolution. We did quite a bit of work led by Alex Harkis while he was a student in my lab. Now he's a PI at Hudson Alpha, to characterize the sex chromosomes and understand this genetic basis of sex determination in garden asparagus. And we did this by sequencing a double haploid male and a sibling female, and comparing their genomes with each other, identifying the bit that was co-segregating with sex, and identified this locus where there's about a megabase male-specific region or Y-specific region. There's also a X-specific region that deserves more attention, but focusing on the Y-specific region has two genes that are responsible for sex determination. And we determined this using with knockouts. First, this TDF1, actually, when we saw that this was in this region, it gave us a hint. This is a MIB factor, which had been implicated before, as I'll talk about in a sec, in pollen, anther and pollen development. It turns out that the other gene that's responsible for female suppression in male plants, so there's two things going on in these male flowers. Female development's being suppressed, and we've got a gene that's necessary for male development. They're together, they're missing on the X. And so, but the female suppressor is this DUF domain of unknown function gene, which we call suppressor female function asparagus. As I said, we knew by just looking at the annotation of this TDF gene that it may well be involved in promoting male function, and not having it in the females would make it so they wouldn't be able to produce pollen, fully functional anthers and pollen. And through some EMS mutagenesis experiments, we verified that we got the same thing going on. That it was found in Arabidopsis. We have the same thing going on in asparagus. You knock this gene out, you can convert a male to a neuter. And so we've got a two-factor sex determination system. It's been reviewed in this recent paper by Renner and Mueller. When we knocked out SOF, we were able to convert a male to a hermaphrodite. When we knocked out TDF1, we were able to convert a male to a neuter. And we knock out both genes, we're converting a male to a female. Now, a current student in my lab, Phil Benz, is doing resequencing of a bunch of other asparagus species, dioecious asparagus species in particular, and putting that into a phylogenetic context. And interestingly, his work's backing up some work that had been done earlier with just PCR, showing that a subset of the dioecious species of asparagus don't have TDF1 as sex linked. It's autosomal, and so both males and females have TDF1. And one thing that we're exploring right now is what's going on with these species. They have the female suppressor that is male specific, but they don't have TDF1. What is promoting male development in those species? And so we're working on that. We also, in collaboration with Alex Harkis and Sarah Carey, have sequenced and analyzed this other dioecious species native to the Mediterranean area, Asparagus horridus. It's dioecious, but it has a completely different sex termination system. The sex termination region is on a different chromosome, and none of the genes that are sex-linked in Officinalis are sex-linked in that species. So that's another thing. So going forward with this, from the last common ancestor of all of these, we're able to see independent lineages of sex chromosome evolution, and see what's going on. How do sex chromosomes evolve from a common ancestor, and how do they evolve in parallel? Okay, next vignette I'll talk about is work that we've worked on CAM, cressolarian acid metabolism, a form of photosynthesis or strategy of photosynthesis that has been, it seems to be an adaptation to increased water use efficiency. And if you remember from your intro bio courses, what CAM plants are doing is they are opening up their stomates at night, pulling in carbon dioxide, then doing primary fixation using carboxylase, keeping that carbon in this 4-carbon acid overnight, and then during the day, stripping off one of those carbons with a decarboxylase to do photosynthesis during the day with the stomates closed, and therefore not losing the water to transpiration. So CAM has evolved multiple times across green plants and flowering plants, and so we're really quite interested in understanding what are some common themes there, and how in the way that it's evolving. Similar to C4 work that others are doing with C4. The evolution of CAM seems to be clustered in various parts of the tree. For instance, here we're looking at agaves, yuccas, and their relatives. And work done by Carolina Hayduck. Oh my God, that's really irritating. A former student in my lab, now at the University of Hawaii, now on her way to the University of Connecticut. She determined that there's both physiological and anatomical traits that seem to have primed the species in this clade for, given predispose them to the evolution of CAM. We have three independent origins of CAM in this group. Interestingly, the primary, as I said before, the enzyme that's doing the primary fixation of carbon, of atmospheric carbon at night, PEP carboxylase. Typically, it's this plant-based PEP-C, so-called PEP1, and this is the same PEP carboxylase or homologs of the same PEP carboxylase that's used to make C4 molecules in C4 species, and in the transcriptomic data is telling us that okay, there we go, that in agave and some of these other species, these type 1 PEP-Cs are the ones that are upregulated at night and responsible for primary fixation. Interestingly, though, in this isn't the case in Yucca allifolia, an example of one of the CAM yucca species. Rather, in this bacterial type PEP-C, which is typically a heterodimer in other species, not involved in forms of heterodimer, not really involved in, it hasn't been involved in CAM nor C4 carbon fixation in any other species, turns out to show a transcription profile and actually multiple copies that are in a tandem or a tandem array that seem to be showing evidence of their involvement in that nighttime carbon fixation in this species. Unlike, again, these other independently derived CAM species in the Agavoideae in this group. We're now in the middle of a project working with in a FICUS-funded project being led again by Carolina. We've known that there's a hybrid between Yucca allifolia, the CAM species, and a C3 species, and this hybrid is segregating for various aspects of its CAM biology, and there are some genotypes that do not do CAM at all. They're fully C3, and so we're taking a multi-omics approach to try and understand both the the genes that are and how gene transcription might be influencing this biology, but also looking at metabolites in the proteome to better understand, well, for one thing, what's going on with this PEP carboxylase. Is it forming homodimers, which would be unique, actually? The bacterial PEP carboxylase is forming homodimers to fix carbon, or is it forming heterodimers with one of plant-based CAMs or PEP carboxylases? All right, and now I'll just mention this, talk briefly about this work that we've initiated in the last few years with Jeremy, this Open Green Genomes Initiative. And for this, we're sequencing about 35 genomes across the land plant phylogeny to fill in gaps in the available sequences, available high quality assemblies and annotations across, again, the open land plant tree. So in purple, I'm showing the target species, and we're actually about halfway through and look to phytosome, that we're releasing these genomes on phytosome. There's about a dozen up there now, or I'm sorry, about 6 up there now, but more to come. Okay, and this is a huge project. There was huge community buy-in and to get to launch this project, and of course with all of these species that we're collecting from collaborators from around the world, and the big, large scale analyses we're doing, we've got over 100 co-PIs and contributors on the project, and of course the JGI, Hudson Alpha, and Arizona Genomics Institute team is really integral. They're doing all the heavy lifting as far as the genome sequencing annotation and annotation. Now, this is enabling us to do a number of things. For instance, one thing we've been working on is trying to reconstruct the ancestral genomes by comparing available genome sequences and reconstructing what the gene content and chromosomal structure is for these ancestral species. And we're using some new software developed by David Sankoff, led by this amazing student, Chao-Chi Zhu. Watch out for her. She is really quite brilliant and doing some doing some amazing work to help us through some computational challenges with this massive project. On Thursday, we'll have a publication of the first genome from the Open Green Genomes project, a fern genome, sea fern, you might know model fern species. Often you may have worked with it in again, an intro botany course, learning about life cycles and understanding the difference between gametophytes and sporophytes. So this has a genome around just under 10 gigabases per haploid genome, and we're working with Jeremy and crew. They've just done a wonderful job of taking this massive genome and assembling it into chromosomes and working on the annotation. Lots of interesting stories. It's a very dynamic genome with genome and gene family duplications. And here's just one story that's in there that we've got multiple instances of horizontal gene transfer from microbes, and then diversification of those genes following the horizontal gene transfer. So this is a phenolic acid decarboxylase involved in metabolism of phenolics, and interestingly, this gene's family seems to have really diversified after horizontal gene transfer, and we have evidence of expression of about a third of these gene copies. So they're doing something. So another thing is, by increasing the density of genomes that have been available, we can better understand genome rearrangements that have occurred through the flower and plant evolution. So this is a beautiful figure that from John Lovell's work, working with an assembly that Jerry has put together for ferris, is one of the species that we've added. And we're comparing that to pineapple down here. Pineapple is in a group that's sister to the rest of the Poales, the order in which the Poaceae is in. And so, but we can see that's quite a mess. There's, for one thing, a genome duplication event in the last common ancestor of all members of the Poaceae, and lots of rearrangements between pineapple and both of these grasses, ferris and rice. By moving a little closer, we've got a genome we generated for Joinvillea just outside of the Poaceae, in a clade that's sister to the grass family. You've got a much simpler picture, it's a lot easier to reconstruct and interpret the rearrangements that have occurred both on the lineage leading to Joinvillea and on the lineage leading to Poaceae, and the genome duplication event. So Joinvillea did not have that genome duplication. All the members of the Poaceae have, and so we're able to use this as an outgroup, if you will, to better understand the consequences and dynamics following that genome duplication. And my last little story is work that we're doing. We at one point, so grasses we know have have a duplication of this phenylalanine ammonia lyase gene, and one of the copies has taken on the function of being able to both metabolize phenylalanine and tyrosine. And so we had wondered that folks had thought, well, maybe that gene duplication event was associated with the whole genome duplication event that occurred in the last common ancestor of all grasses. And the genome data shows us very clearly now that Joinvillea has this duplication, and it seems to be a tandem duplication that occurred to give rise to these gene copies. And then that tandem, the tandem duplicates have been duplicated yet again through this genome duplication event. And so, you know, some grasses anyway will have 2 copies of PTAL and 2 copies of PAL. So we've got that pattern that we've determined looking at the by looking at the genomes, and but work done in the lab of Hiroshi Maeda at the University of Wisconsin, particularly beautiful work by Yuri and Bethany, 2 postdocs in the lab, have characterized the function of these genes following the duplication event. So the TALs are able to the PTALs are able to again metabolize both tyrosine, so they have TAL activity and PAL activity. So following this duplication event, the TAL copy has taken on this, expanded its function. And Bethany and Yuri working together have combined molecular evolutionary analyses and site-directed mutagenesis to identify the amino acids that have really been key in this expansion of function. All right, so that I'm going to leave it there. I thanked many of my collaborators through the talk, and I want to thank you all for your attention. Be happy to take any questions.
Thank you. We got time for one question. Is there somebody in the audience who'd like to ask? Thank you.
Thank you, Jim. So you built these huge gene trees across 1,000 transcriptomes and pulled out the single copy genes. How do you think having multiple whole genome duplications in there and selecting on the genes that end up as single copy influences branch length, for example? Because if you went under a whole genome duplication and then you selected just lost one, maybe that would be a conserved sequence. I don't know. I was just wondering what you might think about that.
Well, there are a lot of issues there. So, so these genes are, you know, there's been constant gene duplication and multiple genome duplications that have occurred throughout the history of this of Viridiplantae. And these are genes apparently, because we see that they're generally maintained in single copy in everything from algae to flowering plants. So they're under some sort of selection apparently to maintain a single copy. And so there's definitely a bias in our using those genes to try to reconstruct relationships. You know, both Tandy and Siavash Merab, the 2 computer scientists that I mentioned helped us out with the initial analysis, have developed new analyses that are able to use multi-copy gene families. So it's simultaneously reconciling to the gene trees and species trees and estimating the species tree. And as far as the relationships, those are robust, the relationships we inferred previously. As far as the branch links, that's a really interesting question that I haven't looked at yet, and it's definitely worth looking at. Yeah.